Welcome back to the chapter, students, and in this module we shall be studying recognition sequences, cleavage pattern and agarose gel electrophoresis. Now let us begin with the module and take a look at what are recognition sequences. Recognition sequences are also called as restriction sites. But what are they? Recognition sequences or restriction sites are the site where DNA is cut by REN or restriction endonucleases. So let us take a look at this DNA molecule. Here we have highlighted a small nucleotide sequence. This is our restriction site or the recognition sequence, and this is a 4 to 8 nucleotide sequence. It is also palindromic in nature, and when a restriction enzyme cuts the DNA, it will cut both the DNA strands simultaneously, and ultimately it will be done by cleaving or by cutting the DNA within this site. The cutting or the cleavage of the DNA will be taking place within this 4 to 8 nucleotide sequence, and it will be done by restriction endonucleases. And restriction endonucleases are highly specific. So each REN is highly specific, recognizing only one restriction site. So remember that, students. Now let us move ahead and take a look at what are palindromes. Here I have a word Malayalam. Now, can you see that Malayalam, when read from left to right, is still Malayalam, and when it is read from right to left, it is still Malayalam. So when a word or when a term read the same in both the orientations, it is termed as a palindrome, or when the reading frame is kept same, it is termed as a palindrome. And our recognition sites are also palindromes. So here I have a DNA molecule, and this GAATTC nucleotide sequence, when read from 5' to 3' direction on both the strands, reads the same. So this is a palindrome, and our recognition sequences or restriction sites are palindromic in nature. Now let us move ahead and take a look at cleavage pattern. Now what is a cleavage pattern? Remember what are restriction fragments? So the type of restriction fragments that we get after the action of restriction endonucleases is termed as cleavage pattern. Here I have a DNA molecule and I cut it with a restriction enzyme. However, these restriction enzymes require Mg2 plus ions for their action, and when they cut the DNA, we get a pattern of restriction fragments which is termed as sticky ends. And sticky ends are nothing but single stranded ends of the DNA molecule. So this is the cleavage pattern. Now let us move ahead and take a look at this single DNA molecule. Now what you see here are multiple restriction sites. So when we have multiple restriction sites and a restriction endonuclease acts upon it, we will get multiple fragments. So presence of multiple restriction site will give rise to multiple restriction fragments, and whenever a DNA molecule or whenever a particular DNA molecule is cut with the same restriction enzyme, it will always give me the same number of restriction fragments. So remember that, students. Now let us take a look at the action of restriction endonucleases, or how the restriction endonucleases actually work when they cut the DNA molecule. So here I have a DNA molecule. Now let us take a look at that small portion. So here I have that nucleotide sequence. What do you see here? You see hydrogen bonds, and you also see phosphodiester bonds. But what or which bonds do restriction endonucleases cut? Restriction endonucleases cut the phosphodiester bond. But how do they do it? They do it via the hydrolysis of phosphodiester bond. And now, as I said earlier, both the DNA strands will be cut simultaneously. So here the hydrolysis of the phosphodiester bond took place, and both the strands of the DNA were cut simultaneously. So cleavage of phosphodiester bond is the method or is the process by which the restriction endonucleases cut the DNA molecule. Now let us move ahead and take a look at agarose gel electrophoresis. Agarose gel electrophoresis is a method of separation of restriction fragments where the DNA fragments run from negative pole to positive pole under the influence of electricity, and we get band patterns of DNA fragments. So what you see here are nothing but the DNA bands. So this is the band pattern that is observed in agarose gel electrophoresis after separation of restriction fragments or DNA fragments. So in this module we studied restriction sequences or recognition sequences, then cleavage pattern, and we studied agarose gel electrophoresis. And in the next module we shall be studying DNA library and genomic library. So till then, take care, study well, and bye bye.